Hello to everyone watching and listening, and welcome to the 7th episode of the BME podcast. For some reason, we just can't stop bringing in super incredible guests, as today we have Roser Sanchez-Todo and Nacho Moragues, R&D Director and Customer Service Engineer at Neuroelectrics, respectively, which I think Neuroelectrics is one of the most famous startups if you are a biomedical engineering student in Barcelona, and for good reason. And hopefully today we'll learn about all the cool things that they do and also their experiences. My name is Gonzalo Plaza, and today I am accompanied by very good friends, Adriana Royuela and Ana Barredo. How are you doing, girls?
Hello, super fine. I'm expecting to start the episode.
Yeah, super interesting.
Yeah, me too. So excited.
So why not start introducing our guests then?
So for those who don't know Roser is an alumni of our same degree, the Biomedical Engineering degree here in UPF. He also specialized in computational neuroscience, doing a master, and now she's the Research and Development Director of the Modeling Department at Neuroelectrics, while she's also studying a PhD about digital twins to model the Alzheimer disease.
And joining us today, we also have Nacho Moragues, which he's also a biomedical engineer graduated, but at the Polytechnic University of Catalonia. And currently he's doing a master degree in data science at the Open University of Catalonia. And he started at Neuroelectrics as a customer service, but now he's currently in the sales department, also as a sales manager. And 2 years ago, if I'm not wrong, he started also as an associate professor at the Polytechnic University of Catalonia. So first of all, welcome you both. We are so pleased to have you here. How are you? What are your expectations for today's episode?
Well, I don't know. I'm ready to be surprised. So feel free to ask anything. I'm very happy. Thank you very much for inviting us here.
I'm great. Thank you for this great presentation. I guess that after this amazing presentation, a bit of imposter syndrome. But, it's always nice to have or to know that actually Neuroelectrics is famous among students. I know we are like celebrities of the biomedical engineering.
Sort of.
Yeah, totally. So some years back, you were both biomedical engineering students that had the opportunity to start working in Neuroelectrics through your thesis in Roser's case and through an internship for Nacho's case. So tell us, what is the aim of Neuroelectrics as a company, and maybe more for Roser, how were those initial years back there in 2017?
Mm-hmm. So the mission actually is to improve patients' lives of people that's living with brain disorders, delivering personalized stimulation therapy anywhere, anytime. How? So with a solution that, spoiler, it's a medical device. Also, there's some software side that Roser will comment later, but solution is a non-invasive, non-drug option for patients whose disease has not been controlled by drugs or by surgery.
Yeah. So it's an alternative treatment, basically, and we'll tell you more about it later. But the main thing that we focus on is to personalize the treatment, because we know that every patient is different, so it's very important to have all the data from the patient and have a personalized strategy. And I've been there since 2017. Doing my internship and my thesis there, and you can see how happy I am because I'm still there,
And it's trapped. Seems like a good decision.
I'm trapped totally, yeah, because I also did my master's there and now my PhD as well with Neuroelectrics. So of course I'm super happy.
Your second home?
It's my second home. So between UPF and Neuroelectrics, they are both like my dad and my mom, they created me completely.
That's so great. And also that you have the opportunity to do your PhD at the same time.
It's amazing. Yeah. So in our department, we are scientists. So we value a lot research and the students that can grow inside our department as well. So we have many master theses this year. We have 3 of them of current employees, and my thesis and other PhDs going on. So it's huge. But this is new. Like when I started in 2017, in the team that I'm at, there were just 3 people, counting myself, And then we've been growing a lot, and we arrived to be 20 as a department. So it's been like a huge growth right now.
And was the vision the same in 2017 that it was now?
Yeah, totally. But now we can say with more ground.
Yeah.
Before it was like, personalized treatment, network mapping, and I was like, yeah, we do personalized treatment. Like we can be more sure and more confident about what we do.
Yeah,
It's amazing.
totally. I think everybody watching at home is looking at this skeleton here, well, this person here with this little helmet. Why not tell us about your medical device, right? And if you want to, maybe give us a more insight about what you were saying about like refractory focal epilepsy, how this is treating it, and a bit of the framework, how you're collecting the data, then modifying, and maybe we'll try to dissect each one of the sections maybe as we go on.
Of
Right.
course.
Yeah, we
You want
can
to
do
start
that.
showing the...
Yes, I think they can see it.
Yeah.
But so Neuroelectrics actually is specialized into two hardware solutions. Roser will talk later about the software side. But this is the Starstim. So let's say in Neuroelectrics we have the Enobio, which is a wireless EEG device, widely used actually for the clinical and the research, for diagnostics, BCI, neuromarketing.
So it's a recording device, right?
It's a recording device, yes. EEG, Actually, mobile solutions for recording the brain, we don't have many because you cannot take someone walking with an MRI or with a CT. You have to do always EEG or FNIRS, which is Fear Near Infrared Spectroscopy, if I'm not wrong. And, this is Enobio, so only recording, as we said. It's a medical device in Europe. We have the CE mark. For the FDA, we also have the clearance, and we're working for the clearance in Mexico. Every country has different regulations, but it can be used to diagnose because it's a medical device. Regarding the Starstim, which is a very similar device, actually they look the same. It's hard to distinguish even
Well,
from...
in the Enobio, you can say that here you just see some electrodes. Enobio is like full coverage of electrodes. If we have 32 channels.
You have up to 32 channels because you want to record the entire brain areas. Actually, maybe if I describe it right, because not everyone is watching it. Okay, so
maybe
in a
like some
in
background
simple
or
terms, yeah.
something.
the device, actually for you, I can also describe it. It's a neoprene head cap with some positions. These holes that you can see or serigraphy, it's the coordinate, an international coordinates. This is the 1010 system. Each of them is above one important brain area. And this is used for EEG. You place the electrodes over the areas that you want to record, and for stimulation, because it's like a coordinates, to locate all the brain areas. You also see that there are some cables on some positions. These are the cables connected to the electrodes, and usually you put gel to create a conductive path to record, or in the case of Starstim, to stimulate.
Very simple question. Can I
Okay,
stop you
go
right
ahead.
there? Can you reach subcortical parts of the brain, or is it only for like the nearest part to the skull?
So,
I can help you with that. Yeah. So you reach very well the cortex, so external part of the brain. You can also play with network information in order to get to the midbrain,
Okay.
but it's very it's in the research right now.
Okay, I see,
So
I
we
see.
know for sure that it reaches the cortex. There are studies that are studying how to reach intracortical regions, and also through a technique that is called temporal interference, which is that you apply two different frequencies, very high frequencies with a difference of two earths between them. So you create like a resonance phenomena in the middle areas of the brain, and you stimulate there at this frequency. So this is it's a very cool technique, this temporal interference, that you can reach subcortical areas.
Just to remark that is always non-invasively.
exactly.
Yeah.
Yeah, the electrode. Well, the only invasive that you get is with gel at some patients, but yeah, not invasive
You
at all.
take a shower and that's
Exactly.
Yeah, it's not.
Or even with a tissue. Yeah, you put gel inside these holes, Okay. Let me try with another one. Okay, this one. You see, you put the gel inside, and you can have clear view of the scalp, so you can ensure that you are creating a conductive path. So yeah.
And the hair does not.
It doesn't matter because
okay.
you can part it and you clearly see
Yeah,
the
because
scalp.
it's pretty small.
Yeah.
There are different types of stimulation. You can do direct current stimulation, which is a constant current. So you have alternating current stimulation. This is actually what it's used for Alzheimer's, right?
Exactly.
Because for depression and epilepsy, you use direct current stimulation. Well, there are a lot of theories, but one that works is cortical neurons are perpendicular to the scalp. So if you stimulate there, the electric field generated is actually parallel to those neurons.
Yeah. So these neurons are called pyramidal interneurons, and they're the most popular excitatory neurons in the brain, and they have a geometry that is very elongated. So you can polarize it very easily with an electric field, because then you move the charges inside this neuron around, and that's the mechanism of action of this electrical stimulation.
So you have Enobio, that is basically for recording EEGs, for diagnostic, and you have Starstim that can also apply the electric field to the brain. And and if I'm not mistaken, this has also been given FDA approval for
Not
epilepsy.
yet.
No.
We are actually focused on epilepsy and depression. Actually, 2 years ago, we ran a clinical trial. It was a pilot clinical trial for MDD, which is major depression disease. The results are published, if I'm not wrong.
Yeah.
You can check that. There's a paper.
I have the paper open here.
Yeah.
And we are right now in the middle of a pivotal clinical trial for the FDA clearance. And pivotal means that multi-site, so there are sites in the US, sites in Barcelona, actually, Vall d'Hebron, Hospital Clinic, Hospital del Mar, and so many that are running our clinical trial to prove efficacy for this specific condition, which is refractory epilepsy, with one focus for patients that are drug resistant.
Yeah, and the pilot that we did some years ago, both the epilepsy and the MDD pilot, they proved like amazing results. Of course, it's open label, there is no sham condition, so we cannot control it with the sham, but they were super promising, so we got to do this pivotal, which is, these pilots were with 20 patients, amazing results. Now we moved to 200, so, and there we can even prove the efficacy with a sham controlled and all these things.
Okay.
So we're working on it. We will get it.
Yes.
We will get this.
You start seeing like an impact of your solution.
Yeah, of course, and we get amazing feedback from patients that they really want to continue. It's a pity that right now we cannot actually target them because they finish the trial like, okay, give me more, and doctor is like, I'm sorry, this is just for research, you cannot ask for more, and they are asking to be involved in other trials as well. So it's
Maybe
amazing.
Just as a curiosity, why did you choose patients from different areas, like so apart from each other? Because you have like not enough patients here in Spain or Europe that have this exact condition, or because you want to generalize better, or?
Yeah, it's very hard to get patients in general for this, mostly epileptic. Right now, let's focus on epilepsy, no? Yeah. So a patient comes in, and there are different levels of hospitals. There are hospitals that do not have the technology to do a clinical trial because they cannot have all these regulatory aspects uncovered. And there are a lot of patients that go to these hospitals, so you cannot access them at all. Then you move to more professional hospitals. And there the patient goes, have a drug, 30% of them do not work. So they enter into a new whole realm, like, okay, so drug does not work, I prove another drug, I prove another work, still doesn't work. Okay, so let's do surgery. So they implant these electrodes, and we are working with patients that have passed so many steps, and they may end up going into surgery. But there are some that the surgery is like in an eloquent area of the brain, so they cannot remove the area. So it's like, okay, so this is a great solution for them, but this is a percentage that it's low. It's big, okay, so in the general public it's huge, the amount of people that are in this stage, but they're very hard to get. So it's very important that we get as many hospitals as we can and as many patients as we can.
And with these patients that they have this condition, that this epilepsy just won't go away, right? To be clear with the terms, you are not curing epilepsy per se, you're treating the symptoms of the epileptic attacks, right? Or the epileptic seizures. Sorry.
Yeah,
right.
Yeah. So epileptic seizures are these huge discharges in the brain, and what we do is try to inhibit with this stimulation. So depending on the polarity of the current and the area that you're targeting, you can inhibit or excite with the DC currents. And in epilepsy is what we do, and we play a lot with this plastic mechanism. You know that the brain is plastic and it learns. It's like a muscle. If you go to the gym, it will get bigger. So if you get stimulated 5 times a week, it will get better results.
But in which sense do you stimulate it? Like, is the area, I don't know, I'm just guessing. Is the area like super depressed and sometimes it just spikes and you want to balance that, or it's just spiking all the time? How does this area work, and how are you exactly modulating that?
Yeah, so there a huge, big range of epilepsy and epileptogenesis. Okay, so it can be by just a kid usually that has epilepsy. So by birth, you have it. For some mutations of the brain, it can be because you get
A lesion,
or just because the cells in this region are a bit scrambled, so they did not get things or orientated right. So there are some inconsistencies there. And the way to target these patients, specifically with the stimulation, right now it has been always the same. So we take the focus, which is where the seizure originates, which this is also another thing that I didn't say before. So there are focal epilepsies and generalized epilepsies, and we just target the focal epilepsies because we take this focus where these epilepsies start and we try to inhibit that area. But this is a solution that we're currently working on that we don't know. I mean, it has good results, but not the best. Okay, so we accomplish like 50% seizure reduction, let's say, but we want to have more. And why? Maybe because the cells that are in that focus are not orientated right. So even if we stimulate them, there is no solution. So right now we're working on another product, another type of stimulation that takes into account the network itself. So not just the bad area or the unhealthy area, but the full network that's where the seizure spreads as well. So we're trying to move a bit this approach.
Because, excuse me, Anna.
No.
Because, no.
Go.
Because with the current product, I'm assuming you create a general model of the brain and you just make the patients put the helmet on and you know where the focus is of the epilepsy and you stimulate more or less in that area. But now you're trying to go to personalized networks of each patient's brain. You have not tried that yet. That's what you're studying.
Yeah, kind of. The two things. So right now the pipeline where we take into account just the focus can also be personalized. Okay, so we have the MRI of the patient. So the doctor comes in, they have a tool that they paint where the focal target is, and they send us the target and the MRI. So we build this 3-dimensional model of the patient. We optimize the stimulation using these FEM models.
Okay, it's optimized for the patient.
It's optimized for the
That's
patient.
actually a head model because we also have the skull and the scalp thickness. So it's a head model actually.
So you see it in 3D and you see the patient's face. I mean, we have to deface it for anonymization, but if we could not do that, we would see the patient's face and we have the head there geometry. But right now we want to improve with the network, so we need more data. So for now, we just the MRI, but we will start another pilot study in 2 months, I hope, that we start in 2 months, that takes into account more data, which is also the diffusion MRI of the patient, so how each area is connected, and also the SEG data, so we know where the seizure actually spreads, not just the focus, we also know the network, So we need a lot of data, then we personalize this model for each patient, and we try to replicate this network spread of the seizure.
And just talking about models, because you say we are modeling the brain, but I guess you're not modeling every single neuron of the brain. So are you thinking like a bunch of cells, or what are you exactly modeling?
Exactly, so it's
no, actually, if you explain it like the Metro of Catalonia, I like when you make this comparison because...
Exactly, so I will answer the question, but it's like the network thing is an analogy as the Metro of Barcelona. So if the focal seizure or the focus is in Plaza Catalunya, it will spread so easily. So the diffusion MRI, it gives us this information about the brain. Or if it's in the hospital, it starts the seizure in the hospital, in the airport, let's say, that is super not very good connected by Metro. It will just, spread a bit more. So it's crucial to have this data of the patient. And what we model is what we call a mesoscale model. So it's not a microscale. It's like in the middle of the full brain and a single neuron. So it's average activity of populations of neurons. It's called the neural mass model. But in order to integrate the knowledge of how the electrical field affects to these neurons, we use microscale models. So we use like single neurons. We try to move the electric field around. How does it interact with each different population type? And then we integrate this knowledge of microscale into a mesoscale that is more computationally acceptable to do a full brain. We cannot model each neuron, but if we take averages easily, it's easy to model computationally speaking. It's cheaper.
But then you cannot introduce still all this modeling at the day-to-day practice, because I think that computationally is costly or not.
Yeah, so the first models that I was telling you about, where we just take the MRI. Right now we have optimized this with artificial intelligence, so it used to take 1 day and a half, okay, to run this optimization, and right now it takes less than 5 minutes. So we have reduced it a lot, and we could use it in the day-to-day life. So we need the MRI. Well, we can do template head, but ideally MRI. The other models that are more computationally expensive, because we model each population of neurons, this takes longer. It can take right now between, I would say between 2 hours and 2 days if it goes wrong. Okay, so they are not so robust with this super research, and we will start now with this clinical trial, but this is the margin. But at the same time, with epilepsy, we have this margin because we need to record first the activity. So they put the SEG electrodes in the brain. They are in the hospital for 2 weeks or so with these SEG electrodes, and we have these 2 weeks to plan the intervention. So even if it takes 2 days, we are super chill.
That's fine. And I think, as you mentioned, like whatever it costs computationally, you can solve that in a certain amount of time. Even with computers, the computers get better every day. So that's not a problem. What I see as a problem with the super personalization, and maybe it is not, so correct me on that, is that you need to take many images of the patient, and that is costly for the product itself, or for the. How do you see this? Are you actually taking into account in the business model or whatever that you have to take so many images, and the hospital has to take as many images as you need for you to run the models and blah blah?
Exactly. So this is a great question that you're raising, because right now we are designing a product that is based on Enobio and Starstim only. That means that we just need EEG and stimulation. But in order to build this model and to be more informed, we need a lot of data prior. So we are using all these studies in order to develop what we call a Neurotwin, which is the fancy name to a computational model of the brain, okay? But we need to do these super costly trials to have as much information as we can about seizures, how they spread, how they not, and then try to simplify it with EEG. So can we get the same results just with EEG and removing all this data? And we will see that hopefully in 1 year and a half, that we will have the data from the trial, and then we can prove that it's actually the same with EEG.
And even with only ECG, you could locate the focus of the seizure, or that's the idea?
The idea, yeah. But if you see the story, like if you see all the people that have tried to localize the focus with EEG, they have failed. Okay, so it's not easy. It's super
Doesn't
hard.
look like.
It's not easy at all. So also, if you try to localize the activity of the brain just by the electrodes, you just have here like 32 in this case. Okay, there are high density ones, but imagine you just have 32 electrodes, and you have a source or a dipole that generates activity in the middle of the brain. Okay, so it's like you are in a party, you know, you have many microphones in the ceiling, and you start talking, you know, and many people start talking. So you're in a party. How can you localize each person in the party with the microphones in the ceiling?
Yeah.
Imagine that there are more people than microphones. So this is the same analogy in the EEG. So it's very hard to localize where this is. There are many techniques, but none of them is the golden standard yet. Okay. So it's not easy.
So your idea, for example, would be to also use ECG to locate maybe how they spread, or that's like too hard and you would only stand as a general pipeline?
Did you say EEG or SEG?
Like the electro...
The
EEG.
The EEG, yeah. Sorry. So this is the goal right now.
Okay.
Right now I see.
So like detect what microphone and which microphones around are also detecting that sound.
Exactly.
Oh.
And I find that incredibly difficult, and if you do that, congratulations on it.
Yeah.
But we hope that they do it.
No, of course, I'm not saying they don't. I'm not hating here.
We will do.
It's complex. It's complex.
Are you also trying to infer the connectivity of the brain from that?
Not the connectivity. I mean, you can.
Like the same information from the DMRI? Are you trying to do that with the EEG as well?
No,
Is
not exactly like this. No, there are different types of connectivity. So there is the structural one, that is the diffusion MRI, and there is the functional one, which is the one that you get with functional MRIs or with EEG. And the whole field in computational neuroscience in the past 20 years have been focusing on trying to link which is the relationship between structural connectivity and functional connectivity. And You know, we need modeling, we need a lot of things, but this is the field where we're working on to try to link these two. So it could be possible to do that right now. I don't know anyone that has
That would be so damn cool.
And
No,
maybe,
because it's so cheap. I mean, Jesus.
yeah, exactly.
yeah. And for example, when you try to applicate this in Alzheimer or depression, you would use like the same pipeline, or like the way you personalize this model, or it would be different? Because you said that how you like put these charges in the neurons would be different if they are like continuous or alternates. But also this Neurotwin would be different in some way.
Mm-hmm.
It is, no? Because for Alzheimer's, you use alternating stimulation protocols. You know, they alternate. It's not like on epilepsy that you have one electrode that delivers negative current, so you inhibit that area.
Yeah, and on Alzheimer's, the mechanism behind is completely different. So in epilepsy, you have these huge discharges in the brain. In Alzheimer's disease, there are a cascade thing happening, so nobody even knows how it originated, okay? But there are some cells that are dying, that are in charge of oscillating very fast. So they create these gamma frequencies in the brain that are around 40 Hz, okay?
Mm-hmm.
So what we do is to try to inject these 40 Hz externally into the patient and see if the neurons can be entrained, which means that they oscillate at the same frequency that you externally inject.
Mm-hmm.
And this is what we do in the case of Alzheimer's disease.
In the effort of?
In the effort of making the neurons oscillate in the frequencies that are in charge of cognitive performance, which is this gamma.
Mm-hmm.
So we're trying to gamma, which is this 40 Hz, is related to cognitive performance and memory tasks, let's say, if you do some research. And then we're like, okay, so let's try to enhance it, you know, and even in healthy participants, they have done this 40 Hz stimulation, and you can see that they have better cognitive test
performance.
Yeah.
Studying,
You're like, okay.
Are you actually, because I read one abstract, because many of your papers we cannot read. Please, UPF, pay some more journals. You were talking about animal models suggest that these 40 Hz induce gamma oscillations that reduced amyloid beta and p-tau depositions, which for everyone, these are two biomarkers hallmark of Alzheimer's disease. Mm-hmm. Have you actually tried this and seen any results on the because that would be even treating Alzheimer's in some sense, you know, you're avoiding the deposition of these plaques, right? So
this is that is a very famous study, which I guess is the one that you're looking at, which is in the United States with, I don't know the name, I cannot pronounce it, I'm sorry, where they stimulate at 40 Hz and they remove in mice this beta amyloid deposition, but this is in mice data. Yeah. And there is Buzaki, which is a very well-known scientist in the field, that kind of rejects completely this idea and say, okay, 40 Hz stimulation does not reduce the amyloid plaque. So it's like, and this paper was very...
So he said no to his own research?
No, the initial research was in 2016 and from a lab in the MIT, I think. Uh-huh. And then Buzaki came 1 year ago and said, okay, this does not work, so revise your experiment. So this is like a super controversy field, mainly because there are a lot of Alzheimer's studies that have already tried this 40 Hz stimulation and claim that it works. And we will find out in our own research. So right now we're doing a clinical trial in a project that is called Neurotwin for Alzheimer's disease, where we actually collaborate with Gustavo Deco and Jordi García Ojalvo.
Oh my God.
Shout out to
yeah.
It's very cool to work with them. So we're collaborating with them in this clinical trial. And we will have the results next year. So we are actually having patients in Rome that are stimulated at 40 years using this. It's a different type of Neurotwin, but these Neurotwins, let's say, and we will see if it works for us.
We're doing this episode so early.
But then, just to know, the patient is supposed to be all the day with the cup?
Good question. So for the Alzheimer's study, they are going home with a home system. So there's a tablet that has the instructions. It's an app, essentially, that tells the caregiver, so usually a family member or maybe a nurse, how to mount the system and on which positions. I don't know if you can see this head cap, but it has colors, so it's very easy to mount. I mean, you know that the blue cable goes to the blue pin. So this way, especially on Alzheimer's patients, that they are elder people, they don't have to go to the hospital every day because the stimulation usually is daily or weekly.
Yeah, and in this pilot that we're doing right now, it's 2 months of stimulation daily, so 5 times a week, 1 hour of stimulation. So every day for 2 months, you need to sit down, you need your family member that puts this cap on, you have your tablets. It's very easy in the sense that you don't need to move to the hospital, which is what happens with epilepsy. And you just get 1 hour stimulating home. You can watch the TV, you can do whatever.
You could even go walk, for example. Or not?
The problem is the Wi-Fi.
The problem is the
Okay.
Wi-Fi. Although for epilepsy, we're using SIM cards.
Yeah, that's
But
true.
you know,
Yeah, that's a
Oh.
good point. You could go walking if you carry the tablet very close to you. And,
exactly.
yeah, because some patients are like, yeah, I'm going to the toilet, when they are so far away from the tablet.
Yeah,
Okay,
yeah.
just do everything or just bring the tablet with you.
But yeah, you can move, so you have freedom. You could cook,
Yeah. Sí, sí.
And are you actually selling them? Like, I can buy one of those or not?
So the Enobio, you could buy it, but I mean,
I'm not rich, but
it's not that expensive. I mean, it's compared to many EEG devices. You can buy an EEG device, but it's true that it's usually related to a hospital or to a research group. And Starstim, it's only for research right now, so you cannot diagnose or treat anything actually.
Yeah.
Yet. But yeah.
But we will make it accessible soon.
Yeah. We finish the clinical trials.
Yeah.
Actually, next year and a half, more or less, we will be the first company that has a certified medical device to treat refractory epilepsy.
Exactly.
And well, how, like, when you started this device, for example, well, as you said, it was just for recording, but then you intended to apply it for this specific condition. But the Alzheimer and the depression came after, or it was like all the three conditions at the same time? Like, you saw an opportunity of market or something, or?
Well, I mean, I think that we grow with the market and the research and academia. And, well, there's evidence that stimulation works on these diseases. So the opportunity grow with the research and the science knowledge.
And we have tried other pathologies as well. So also the
there's
ADHD, there is the stroke as well. So there are other things going on, but the most promising ones right now in the field are epilepsy, for sure, and depression. Alzheimer is very like pioneer research, so don't trust it so much yet. But with epilepsy and depression, we can be super certain that this is helping patients and that they want to continue afterwards. So it's very motivating for us in a company to have patients that say, okay, please, can you certify this so I can use it,
Exactly.
So
And maybe for the depression case, like, if you could explain a little bit better, how does exactly what Gonzalo mentioned, that how does this stimulation work for a depressive person? How, like, maybe you inhibit those neurons that are making this person.
what happens in their brain, right?
Yeah, exactly.
You want me to?
Yeah.
Okay. So
There's an asymmetry,
there are
right?
two things. Yeah. One, the gold standard right now in depression, I will tell you first this. So the gold standard in depression right now with transcranial stimulation is that you have a depressed area on the brain that is the left dorsolateral prefrontal cortex. Okay? So this
I won't forget.
this one here in the frontal cortex. And usually when you look at fMRIs, scans of these patients, and you compare them with healthies, you see that this area is depressed. And it's part of a default mode network, whatever. There are some things going on there. The important thing is that this area is depressed. So initially in the field of transcranial stimulation, they were like, okay, let's target this area. And they went all crazy about the left dorsolateral prefrontal cortex. It's like a magic area if you look at it. I'm telling you, it does many things. So they were like, okay, let's stimulate it, and it kind of worked, which is very nice. Okay, so and there is already a certified device that has this electrical stimulation bifrontally at home. So you can stimulate this left dorsolateral cortex. And but right now the field are evolving a bit. And we are like, okay, the depression is not a single area pathology. It's a network thing. There are clusters of patients that have different symptoms. Let's say, so maybe you are more depressed, you don't move so much. Or maybe your mood changes. Or, there are different types of depression.
Yeah,
it's
like
not
symptoms.
just
Maybe if you excite this like motor part, if you have this type of depression where you're like most at bed or whatever, maybe you feel better.
Exactly.
That's what you mean. Okay.
So we are orienting our research. So right now we ask for a grant, okay, in order for them to give us money. Like, okay, we acknowledge that the left dorsolateral prefrontal cortex works but we know there is more and that it doesn't work for everyone. So let's try to find out why. And we are reading papers that are super new. Okay, this is just new things that they like clustering the depression patients in 3-4 groups, and we're thinking, okay, let's stimulate and optimize for these groups independently. So let's have even more personalized treatment with these patients, and also combine it with psychedelics. So right now, psychedelics, we know that it works with patients of depression. So can we combine these two? Because psychedelics also improve the plasticity of the brain. So maybe if we combine them, we can get better solutions with the stimulation. So right now, we're kind of pushing a bit the field on depression because it's a bit...
But it's hard to run clinical trials for medical devices because, if a company... a pharma or a medical device company is sponsoring a clinical trial, we need to hire a company that hires hospitals to look for patients. We cannot directly go to patients and do the stimulation because we know them and we can say, just on the form say that it worked, So, and usually hospitals prefer to give a pill and that's it. So they don't need to learn how a tablet works, an app works. The patient prefers to take a pill, and what they get for the same effort. Yeah.
And also, I guess, whenever you try to move to a new field, you have to hire new people, or maybe you guys are so interested in
with
this
their PhDs, maybe.
How many are they now? Because
20, I think.
Yeah, I think they said 20.
No, I mean in brain modeling, which is our department that is research, it's less than 20 now. We arrived to 20 with interns. Now we are kind of 14, I would say, between 12 and 14. I don't know, because there are many interns. We have a lot of Pompeu TFGs and students here for sure. But
this is just research. The research that they do is cool because, at least from the university, the research that I saw was far away from a real product and far away from helping the society. But the one that they are doing are very close. But this is not all that we do in the company. We also have operations, we have sales, we have production, because we manufacture the device here in Barcelona. We also have an office in Boston. But there's people working, not just publishing papers there.
Yeah, totally. And we need them for sure. We are just another group, we have the engineering department, the regulatory department, the clinical department. So
Exactly.
they are all behind trying to, to make this dream happen, because we are research and we are actually changing a bit our research. So right now I was a research director, now it's research and development director, because right now we're growing into doing a proper product. It's not just a research code that you can say, this is it. No, you need to have a pipeline and prove that you actually did all the tests on your code, that you have everything under control. So we are growing also into this field, but we are not alone.
More business focused, maybe inside research also.
Exactly. And
the product department.
Doing software in a medical device company is not like coding, submitting the change. No, you need to document everything, you need to test everything, you need to validate everything that you do. If you do a new release or a fix, you need to document everything. And it
takes more time to document, I think, than to change.
Totally.
But you are putting this on someone's head. It needs to be safe. It's not because we want it to be safe, which of course we want, but there are regulations and you must comply with them.
They force you.
Exactly.
For a good reason.
For a good reason, of course.
But it's a pain, honestly. But we will make it happen.
So from this shift, now we're going many years back, and maybe you guys weren't even there in the company. We saw, researching, right? That first Neuroelectrics had sort of some research going on with brain-to-brain communication. Where's that gone? Because that looked very cool. Of the concept that when they sent you something, you saw a flash in your head is a bit okay, but it still was brain-to-brain communication. Where has that gone? Is that still a thing, or in this shift to more research and development, that's been discarded?
Well, that's been discarded for this reason, for sure. But main reason is that in Neuroelectrics, we are a medical device company and we want to be in the medical field. And this brain-to-brain communication helped us a lot in order to create the device. So there was a project that is called HYPE, I think, where we were collaborating with the UPF or with other entities. And if you meet Julio, which is the president and the CTO of the company, you will see that he's a crazy scientist and he loves to explore everything, and he's talking about consciousness, brain-to-brain communication. It's very passionate about many other things, but we need to touch reality in the sense that, but we want to help people
Yeah.
So let's focus. And the product department came and was like, okay, this out, this out, this out. And
I mean,
like,
business is business, you know.
see, and prune everything that was not even, I mean, it was a drama for a moment. All the products that we were having for research, it went away, you know. And, but it, I mean, it
But it makes sense, like, you have to eat also.
Yeah, exactly.
Yeah. And well, maybe like the aim of the company, it's not to do like some type of product to communicate, more, as you said, to help patients.
Exactly.
Yeah, maybe it's more for Elon Musk or...
Maybe.
Yeah.
But we continue thinking about this. So even though we don't actually do that for work, let's say, we are, in my team, mostly we're super passionate about other things, and we keep talking about brain-to-brain communication. What is consciousness? Can we actually, you know, so we're asking this essential question. At the end, we're studying the brain. So it's like, this is a super cool organ. You cannot just stop there.
Yeah.
I mean, it's huge what you can do and research in the brain. So if you see some papers that we're recently publishing, it's like, okay, how do you combine this, I don't know, like geometrodynamics is called, like how we model the brain as a landscape, then when you take psychedelics and you do some task, it flattens or not, you know, and how you deal with suffering. And
Psychedelic itself.
yeah. So it's we keep thinking about this even though we actually don't do it for work.
And maybe, well, in this research, they explained that you saw like this type of flash. Maybe if you stimulate like some specific region, you could see like an actual image of, I don't know, a bird. If you touch the region or, I don't know, like identify one object itself. Maybe you don't see it, but you just have that spike of the neuron and you recognize it.
Yeah, right now I think that this is possible with intracortical stimulation. So not
not in the skull.
invasively. I think you can create these images for blind people. You can see numbers as well, and there is research going on that I mean, it's very cool what's going on, but it's also super invasive.
Yeah.
With non-invasive stimulation, since the currents are super low, so it has to be safe as well, and you cannot burn your skin or whatever, you know. So this is barely right now not impossible, because I don't want to think that this is impossible, but it's very hard, I think, and not possible right now in the technologies that we have developed. No.
Yeah, totally. Also, I think going back to something that we were mentioning earlier about the research and business. How do you think being a biomedical engineer can help you in both of those areas, right? Because you guys are both biomedical engineers, but you work in certainly different departments. What are the skills that you learned that can help you in those two, right?
So being in customer support, it's like you deal with tickets, with customers complaining, but it's not like they speak your same language. I'm sure that if you did an internship in a hospital, you will know that doctors sometimes do not speak in a techie way. And, I manage cases with psychologists, neuroscientists, or even linguist people, and they refer to the device as the battery, and you start thinking, okay, so it has a problem with the battery. But no, they mean the entire device, it's a battery, or things like that. So yeah, communication and adapting to who's talking to you. It's what they always say in Open Doors, that biomedical engineering is very interdisciplinary, blah, blah. So I saw it in that way. But, you have to still specialize in stimulation, in EEG, to know them, because then there's someone super expert going to come about EEG, about signal processing, and you need to help him because he paid for the device, he paid for the customer support service. So yeah, more or less.
But then you expect the doctor to use the device itself, and the software is kind of friendly for them to just let them use alone, or you are behind to support them?
So by support, I mean, when something breaks or trainings, especially for the clinical trial, we are taking care of the trainings. We go to the sites, we deliver a training of how everything works. And actually everything is user-friendly. But, on the hospital, there are many apps, many softwares, this machine that works with this app and uses Bluetooth, this other one that uses Wi-Fi. So it's hard for them to know. But yeah, ideally, and it's what we intend to, the software is very user-friendly. You put the patient ID and you get the treatment and how to place everything. So yeah, the support that we gave is as any medical device or as any product,
Totally. And
In Roser's case, maybe.
Yeah, Roser's case, it's maybe more obvious, I guess, but
Yeah.
Yeah, I mean, it's more obvious, but at the same time, I would like to add that, okay, we are biomedical engineer, no? So we have, I would say, confidence. We have the confidence to go into papers that are super bio, and you're trying to understand Alzheimer's disease. So you're talking about proteins and how they form, and the gene expressions, and this APOE, whatever. And you are actually understanding that, and then you're able to go back and explain it to whoever. Well, you go back and you code it, and you are not afraid also to code, you know. So there are no blocks. Being a biomedical engineer is like, no, I can understand this, and then I have the confidence. I mean, I'm not a developer at all, but I have the confidence to go and code something and learn. So, okay, how do I do proper code? So you read, okay, I need to document, I need to type hint, whatever. And then you go and you build your own code. So I think being a biomedical engineer is like about confidence that you can make it.
I think that's the beauty of it. No, love what you said right there.
Yeah.
Love what you said. Completely
Because I
agree.
think one issue that we all have in this degree is that we feel like less confidence because we know less of each subject. Like we know of everything, but so bit.
Yeah.
So sometimes we feel like data science or a biologist or some other person is going to replace us.
No, I completely understand. Like at the beginning when we started doing the developing part, so trying to do a pipeline on how to do proper code and not scientific but more like serious one, it was like, oh fuck, you know, like we need to do merge requests, they need to revise our code. Like we were kind of panicking, you know, a bit because this was not in our field. And of course, if we hire a developer, they would do it like this, you know.
Yeah.
But at the same time, it was like, okay, a bit of learning curve, but we made it.
Yeah,
and right now we have
whatever's challenging is cool also.
Yeah.
You learn
And
so
we
many
learned
things.
a lot. So at the same time, it's like, yeah, that we can make things that we think we cannot. I mean, it will take longer for us, but we will get things much faster.
Sometimes I think I'm just one person. If you need two, it's going to cost you more.
Exactly.
So
Exactly. For us, it's exactly
we have,
like that.
the skills to do it, just that we haven't done it before, I think.
yeah,
Totally.
Yeah,
Totally.
exactly.
So, just to wrap this up, there's been an incredible episode. Really, I would keep talking about this device for many. I have many questions that I haven't even asked. Maybe I'll ask them later now.
Okay, of course.
the podcast will keep going, but not for you guys.
Episode 7.2.
Yeah. Any last advice for the biomedical engineers listening to us right now?
Oof.
I mean, I would reiterate what I said. Like, anything that you're passionate about, don't be afraid of doing it because you will make it. Like all these tools that you're gaining that are super different, they are just giving you confidence to do anything that you want. So just go for something that you really like. And then don't be afraid, just do
I think
it.
we are seeing this in you
Totally.
because like you are talking and loving what you are explaining.
Totally.
Yeah.
I'm very lucky. Like this is passionate. You just find that and just go for it. And I mean, I'm telling you that there will be no blocks because your abilities as a biomedical engineer will make easy whatever you want, honestly.
This is what a teacher that I had at uni, Beatriz Giraldo. She was teaching at electronics and in biomedical engineering. And what she told us is that in the other engineering, when there is an error or something that breaks, it's usually a financial loss, and that's the only importance it takes. But on biomedical engineering, you always have in mind what's at the end, which is a patient's life. And this is very rewarding because what you're doing, you're not coding to make an algorithm that traps people into scrolling more.
Yeah.
You know what I'm saying? You're actually helping.
Great. Thank you so
Should
much.
we stop here?
Yeah,
Yeah.
I think we should stop it there. Thank you so much for being here. It was really, really a pleasure. It was such a fantastic conversation. Thank you, girls, too, for being here. Great episode as well.
Yeah.
And also, thanks to our beloved technician and our today spectator,
Our fun.
which you don't see. Thank you so much for everyone that makes this possible. And see you in the next episode.
Thank
Bye.
you.
Bye.
Bye. Thank you.